So far, anything that's gone on tonight, anything that's been said, anybody need to have anything repeated? You want me to go back over it? Do you have any questions about anything that's been said so far? Sinuses will be hating themselves, and now that I've said that, they're probably going to flood.
Yeah, I know. I think it's the Walters actually starting to work.
I think it was the French fries. I think the French fries have kind of counteracted whatever it is. Yeah. Anybody? Anything you want to go back over at this point? Anything you need to hear again? Yes, ma'am.
Did you have like a name for the, when you said oxygen is stored in the protein called?
Myoglobin.
Yeah, okay. Those two things, did you have like a name for that? Because
No.
you had, it's just like a fact.
It's just a fact.
Okay.
Anybody else want to ask any questions? All right, so on we go with smooth muscle. Oh. Okay.
So this is still smooth muscle?
This is still smooth muscle. One more little factoid about smooth muscle. It doesn't have any T-tubules. Now there are two types of smooth muscle tissue. One type is called single unit smooth muscle. The cells, the individual cells of smooth muscle fibers in single unit smooth muscle are typically connected to each other with gap junctions. Boy, are we reaching back to that early part of the semester. Does anybody remember what gap junctions are?
Isn't that where the no. Isn't it?
The one like this?
Yes.
Oh, okay. The different
I don't
types.
know what your hands were doing. Hers were doing something I understood.
I was thinking about the thermal something junctions.
Desmosomes?
I think so.
Yeah, okay. No, that would be something different. Right. With gap junctions, what we have is little holes, little gaps in the cell membranes, so they're connected to each other with proteins. But there's little gaps, so the cytoplasm of one cell is in direct connection to the cytoplasm of the next cell. Now that we've talked about action potentials, I think you're going to see the beauty of this system. Because when an action potential comes down to the end of a cell, it stops. It can't spark over between one cell and the next. But with gap junctions, if one cell is experiencing an action potential, and that action potential comes down to the end of the cell, it's going to go right on through, and the next cell in line is going to get an action potential. It doesn't have to slow down, it doesn't have to stop, it doesn't have to be regenerated with acetylcholine. So with gap junctions connecting these cells, all of the cells in the tissue are going to respond virtually simultaneously to any kind of stimulus. They're all going to contract at once. All of the cells are going to act as a single cell, hence single unit. So if one cell gets stimulated, they all get stimulated, they all have action potential, and they all contract. We find a lot of single unit smooth muscle in our visceral organs. Not the liver. That's got to go on the test. We got to make that. We got to make that a choice. All right. So things like stomach, intestines, urinary bladder, most of their smooth muscle is single unit smooth muscle, and that works pretty well because that means that the whole muscular tissue is going to contract and squeeze and relax and contract and squeeze all pretty much at the same time. The other type of smooth muscle is called multi-unit smooth muscle. This type is found in the bronchi. Where are those? Well, they're not in the lungs. They're the passageways that's carrying air toward the lungs, which is why I said lungs don't have any muscle, but the passageways do. So in the bronchi, the erector pili muscles, where are those? We talked about those. In your skin attached to the hair follicles, very good. And the internal eye muscles. Now, this makes a good moment to point out to you guys that in the lab, while you're studying the cranial nerves, there are several mentions of external eye muscles. The external or extrinsic eye muscles are the ones on the outside of the eyeball that roll your eye around in the eye socket. They're making the big eye movements. The internal eye muscles that I've just mentioned are actually inside the eye, and they're going to dilate and constrict the pupils and change the shape of the lens so that you can focus near and far. So the internal eye muscles or intrinsic eye muscles are smooth muscle. The external eye muscles that you're learning about with the cranial nerves are skeletal muscle because they're attached to the bones of your skull. Okay. The cells in these kinds of tissues have few gap junctions. So they're not all going to fire off at once. Multi-unit smooth muscle is mostly controlled by neurons, and these neurons form groupings of cells very similar to motor units. So an action potential in a neuron will then stimulate all of the smooth muscle cells in that grouping, but not necessarily all the muscle cells that are present. So these work a little bit more independently, or at least the groups work independently. Did we lose Stevie? Okay. All right. Now comes the big one. Excitation, contraction, coupling in smooth muscle, which is not the same in every point as it is in skeletal muscle.
Is this still included in multiunit?
No, this is all smooth muscle. All. Some things are the same. For example, in skeletal muscle, we need calcium in order for the muscle cells to contract. In smooth muscle, we need calcium for the cells to contract. In skeletal muscle, the myosin heads had to have a phosphate on them in order for them to form a cross bridge. In smooth muscle, the myosin heads are going to have a phosphate on them in order for them to form a cross bridge. So we need phosphates, we need ATP, we need calcium. What we don't have in smooth muscle is troponin. In skeletal muscle, troponin regulated when cross bridges can form and when cross bridges cannot form. It's the regulatory protein in skeletal muscle cells. Smooth muscle cells don't have troponin. They have a different regulatory protein. Also, skeletal muscle fibers typically don't depolarize themselves. Skeletal muscle is not typically spontaneously contractile. Under normal physiological conditions, you have to decide to move that muscle before it's going to be stimulated, before it's going to have action potential, before it's going to contract. Now, of course, there's oddities, but smooth muscle, on the other hand, is spontaneously contractile. The cells will just depolarize themselves and go far off their own action potential without any outside stimulus. So we're going to start with the action potential wherever it comes from, whether it's being stimulated by a neuron or whether it's just decided to do this by itself. We're going to start with the idea that the muscle cell is having an action potential. The first thing that's going to happen is that we're going to open calcium gates in the sarcoplasmic reticulum and in the sarcolemma. The cell membrane has calcium gates. The internal organelle membrane has calcium gates. Action potential is occurring, those calcium gates open. Calcium comes into the cytoplasm of the cell. Calcium concentration starts to go up. These calcium ions, whether they're coming from the sarcoplasmic reticulum or the extracellular fluid, the calcium ions bind not to troponin, but to a different protein called calmodulin. Calmodulin. M-O-D-U-L-I-N.
It's going off the side.
I know, I'm sorry. I can't do anything about the size of the screen. I had a bigger screen and they took it away. I know. Luckily, we had this one in hiding in one of the back rooms, and so my guys put it up for me. Yeah.
I was gonna say,
Yeah.
That was the whole length of it. Yeah, it was really nice. All right, so when the calcium ions bind to the calmodulin, that is going to activate the calmodulin. Now can you see how it's spelled? When calmodulin is activated by having picked up or attached calcium, it in turn activates another protein called myosin light chain kinase. So calcium binds to the calmodulin, that turns the calmodulin on. The calmodulin is then going to turn to this other protein, myosin light chain kinase, and activate it. Now, what the myosin light chain kinase does is it's going to take a molecule of ATP. It will remove the phosphate and attaches that to the myosin head. So the myosin light chain kinase is going to take a molecule of ATP, snap off one of the phosphates, and stick that phosphate onto the myosin head. Now that the myosin head has been phosphorylated, the phosphorylated myosin will now attach to the actin in a thin filament, and we have a cross bridge form. Now, in skeletal muscle, we use the same ATP to power the power stroke. One ATP to form the cross bridge and shift it from a 90-degree angle to a 45-degree angle. Not true in smooth muscle. We need one ATP to phosphorylate the myosin and form the cross bridge. But now we're going to need another ATP to supply the energy for the power stroke. The thin filament will shift from a 90-degree angle to a 45-degree angle, and the thin filament will slide across the thick filament, but we're on this weird kind of spirally pattern, so the cell is going to shorten, but it's also going to bulge sideways. I don't know. It just does. I don't know that anybody's ever proposed a reason why this is such profligate, wastrel sort of use of ATP. Why not use just one and get it done? I don't know. Okay. As long as the smooth muscle cell is experiencing action potentials, the calcium gates are going to stay open. Calcium is going to be high inside the cell, so calmodulin is going to stay calcified. Calmodulin will stay active. It, in turn, will keep the myosin light chain kinase active. So the myosin light chain kinase is going to keep putting phosphates on myosin heads, and the myosin heads are going to keep forming cross bridges and power stroking and sliding the filaments. So as long as the cell is experiencing action potentials, we're going to get the cycling of the thick and thin filaments, we're going to get the sliding filaments, we're going to continue to experience muscle contraction. When the stimulus stops, when the excitation is over, calcium pumps are going to start pulling calcium out of the cell. Some of this calcium is going to go back inside the sarcoplasmic reticulum. Some of this calcium is going to be pushed all the way out of the cell back into the extracellular fluid. But calcium levels in the cell are going to start to go down. And that means the calcium is going to come off the calmodulin. That in turn is going to inactivate the myosin light chain kinase. If the calmodulin doesn't have calcium, it can't activate the myosin light chain kinase. So now the myosin light chain kinase becomes inactive, which means that we can't put any more phosphates onto our myosin heads. Which means we're going to get no more cross bridges, no more power strokes, and the muscle cell will now relax. In skeletal muscle cells, you needed another molecule of ATP to break the cross bridge. If you didn't have enough ATP, you got rigor mortis. Everything just locked in and wouldn't let go. Smooth muscle does not experience rigor mortis because it doesn't take another molecule of ATP to break the cross bridges. We're not making any more, but we still have the last few that were made on the last go-round. There is an enzyme here called myosin phosphatase. that's going to remove those phosphates from the myosin heads. Breaking the last set of cross bridges and releasing the thick and thin filaments from their connections. So we don't get rigor mortis because it doesn't take ATP to break the cross bridges. All we need is the myosin phosphatase, which was there all along anyway.
So I wonder if that's why it has shoot to contract instead of skeletal muscle.
Maybe. But, you know, there isn't always a reason. I mean, the reason is it works. And the cells stay alive, and the tissue stays alive, and you stay alive, and you have babies, and they get the same system. As long as it works and everybody's reproducing and making more, good enough is good enough sometimes. Doesn't have to be perfect, just has to be good enough. Keep you alive long enough to have babies. Darwinian evolution comes. And then if you strap jet rockets to the top of your car, you take yourself out of the gene pool, and we're all happy to see you go. All right, some more stuff about smooth muscle. Smooth muscle cells contract slower than skeletal muscle fibers. If you give a smooth muscle cell and a skeletal muscle cell an action potential at exactly the same time, the skeletal muscle cell is going to contract very quickly, give you that twitch. Smooth muscle contracts slower. It is, in fact, 30 times slower. It'll get there when it's ready. So it responds more slowly, it contracts more slowly, but it can sustain force much longer. Smooth muscle doesn't fatigue nearly as quickly as skeletal muscle cells do. Skeletal muscle cells use up their ATP, and then they start to lose force. Smooth muscle cells take their time getting there, but they can sustain that force for a very long time. And one more compare feature between smooth muscle and skeletal muscle is how these guys respond to stretch. Now, this is always very difficult to get across when we're talking about an intact human body, because typically your joints don't let you pull your muscle beyond its normal orientation. It's very hard to stretch a muscle in an intact human being, unless you hyperextend a joint, and that hurts a whole lot. So most of these studies have been done with skeletal muscle, have been done with muscles that have been removed from the frog or whatever. So if you take the gastrocnemius off the back leg of a frog and you hook it up to little pins, right? If the muscle is just kind of compressed, not oriented properly, and you give it a little electrical shock, you give it a little stimulus, you don't get a whole lot of force out of that contraction. If you start kind of stretching it out a little bit, you get more force, and then you stretch it out a little bit more, and you get a little bit more force. The x-axis here is how far apart they are. And if you stretch it out a little bit more, close to where it would be in the frog, with one end of the muscle attached to the femur and one end of the muscle attached to the tibia, you get a pretty good amount of force. Frogs are really hoppy, right? Boom, get a lot of force. If you keep going, if you keep stretching this sucker out, what you see is after that kind of optimal length where it gets a good amount of force, if you keep stretching, it starts to go down again. The muscle becomes less and less forceful until it gives up altogether. That's because of the sarcomeres. If the muscle is tightly compressed, when you start, the ends of the Z lines are very close together, and you ask it to contract, and it can only go so far. So you don't get a lot of force. As you stretch it out, and the Z lines are further apart, now when you ask it to contract, you get a lot of force. It gives you a lot of oomph, oomph being a unit of force. But if you keep stretching, what you end up with is there isn't very much overlap between the thick and thin filaments. It's hard to form cross bridges into empty space. So if the muscle gets too stretched out, you can't get any force out of it because you can't make any cross bridges. Kind of get that, sort of, right? Smooth muscle doesn't have sarcomeres. Smooth muscle doesn't have Z lines. The thick and thin filaments are just kind of wound around each other in that spiral pattern. Yeah, if the muscle's too compressed, you're not going to get much force. That's the brown line that I drew in here. So if the muscle's compressed, you're not going to get a lot of force. But when you start stretching smooth muscle, the amount of force you get when you ask it to contract goes up. And then you stretch it some more, and it still gives you a lot of force. And you stretch it some more, and it still gives you a lot of force. And you stretch it out some more, and it still gives you a lot of force. And long after skeletal muscle's given up the ghost entirely, smooth muscle will still contract, even though it has been stretched and stretched and stretched. And when you think about where we find smooth muscle, we're pretty glad that it has this capacity. Smooth muscle is in places like your urinary bladder. So Jen here was drinking a 36-ounce drink. Looks like most of it's gone here. I don't think she went to the bathroom. I think she went to answer the phone. But let's say she had to go to the bathroom. Why did she have to go to the bathroom? Because she drank all that drink, and her kidneys were making urine, and the urine was going down into the bladder, and the bladder was expanding to hold the urine. And then it kept on expanding because she kept on drinking, and the kidneys kept making urine. So the bladder expands, and it expands, and it expands. It stretches and stretches and stretches, and the smooth muscle stretches and stretches and stretches. And when you finally can't stand it anymore and you go down to the ladies' room, that smooth muscle can still contract and push the urine out, and you can still pee, even though the muscle was widely stretched. Right? You go to the all-you-can-eat buffet, and you eat, and you eat, and you eat, because darn it, you paid $12.95, and you're going to get every single thing you want. And all of that goes down into your stomach, and your stomach expands, and your stomach expands, and your stomach expands, and it stretches and stretches and stretches. And you can still digest all of that food because the muscles will still work. Skeletal muscle won't do that, smooth muscle will. It is much better at contracting even in a stretched condition than skeletal muscle is. And that works for the organs we find smooth muscle. All right. We have some time. Let's go ahead and get started on the nervous system. I will do cardiac muscle at the beginning of A&P2 when we do the cardiovascular system. And we'll do all the same stuff. We'll talk about compare and contrast. We'll talk about excitation, contraction, coupling, and we'll talk about fatigue and this kind of stuff. But we're going to save that until we get to A&P2. So now we're going to take on our last organ system, our last system for this class. We're obviously not going to have time to get to the endocrine system. I know.
Do you normally?
No.
Oh, okay.
The endocrine system is technically supposed to be part of this class, but I haven't gotten to it for probably 3 or 4 years. It's Well, doing it justice takes time. We talked a little bit about endocrine when we talked about PTH and calcitonin, but it would be wonderful if we could give you a little bit more information because it's a regulatory system. Like the nervous system, the endocrine system controls other systems, right? So it'd be nice to be able to put those two together. Typically what we've been doing in the last couple of years is we run some supplemental lectures in A&P 2 to cover what we didn't get to here in this class. So you will get some, especially if you take me. Oh, goody. All right. So let's talk about the nervous system. The nervous system, like I just said, is a regulatory system. Its function is primarily to control other organ systems. Now, it's organized into two main, I shouldn't say it's organized, we see it as being organized into two main sections. There is what is called the central nervous system, which is composed of the brain and the spinal cord. Brain and spinal cord form the central nervous system. Anything that's not brain and not spinal cord, but is part of the nervous system, is referred to as the peripheral nervous system. Peripheral meaning it's outside or to the sides of the brain and the spinal cord. So we have the central nervous system, brain and spinal cord, and then we have everything else that's the peripheral nervous system. Now the job of the peripheral nervous system is to keep the central nervous system informed about what's going on in your body and to follow its orders. So the peripheral nervous system is then itself separated into two sections. There's what's called the sensory division. Sensory division is taking information in to the brain and the spinal cord. Light, sound, touch, taste, pain, pressure, heat, cold, body position, all this kind of stuff. The brain needs to be informed, the spinal cord needs to be informed. Sensations, perception of sensations, that is going through the sensory division. And the sensory division itself is then subdivided into senses that are coming out of your internal organs, like the liver, and the kidneys, and the stomach, and the intestines. This is called the visceral senses. Now, you're not typically super aware of these. They're not usually reaching your conscious level. You might, when you're in the lab and you're looking at the brain, they talk about how the thalamus is a relay center for sensory input. Not everything that your nerves are telling you about, it needs to be brought to your attention. So the thalamus is getting this and they're saying, okay, that you need to know about, but that you don't. And so some of this is things like blood pressure and how fast your heart is beating and blood chemistry, and you really don't need to know that right this moment. So those are visceral senses. And then we have the somatic senses. This would be things like touch, light, sound, pain, taste. Somatic sensory division. And a lot more of this is probably sent up to your cerebral cortex for conscious awareness than visceral. Although again, not all of it. Your thalamus does edit a lot of this stuff out. One of the things that the somatic senses are doing is they're keeping track of where your body parts are, what your body position is. And typically you're not sitting there worried about where your feet are just because you can't see them. They're there, you know it. Right? Another example of this is your underwear. You're wearing underwear, or if you're not, don't tell me. Let's assume that you are. And most of the day, you're not thinking about your underwear, nor are you aware consciously of the pressure of the elastic and the fabric against your body. But now that I said underwear, your cerebral cortex is queried down to the thalamus. Am I wearing underwear? And the thalamus is like, yeah, can't you feel it? And your cerebral cortex is like, oh yeah, it's crawling up my crack, I know it now. And now the thalamus is like, okay, you're good, right? So when you need to be consciously aware of it, you can access that information coming up through your sensory division. But again, a lot of this doesn't need your immediate attention, and your thalamus will edit a lot of this kind of stuff out. So we have the visceral senses and we have the somatic senses. The other part of the peripheral nervous system is called the motor division, motor meaning to move. And this is the signals that are coming out of the brain and out of the spinal cord to make things happen, to make muscles move, to make muscles contract, to make glands secrete. Things are happening, things are moving. That's your motor division. Now again, the motor division is then subdivided into the part that controls your skeletal muscles, the somatic motor division, and the parts that control smooth muscle, cardiac muscle, and glands. That's called the autonomic motor division, or the autonomic nervous system. This is mostly involuntary subconscious stuff. So changing blood flow when you stand up and leave the room, changing your blood pressure when you're running down the stairs to get to your car before the giant bear eats you. Autonomic motor functions, you're not thinking about it, you don't even have conscious control over it, but nonetheless it is being controlled, autonomic. Now, just to make your life perfectly miserable, and don't blame me, I did not name these, the autonomic motor division, or the autonomic nervous system, is then itself subdivided into two components. These are called the sympathetic branch of the autonomic nervous system and the parasympathetic branch of the autonomic nervous system. Ugh. Now, if you ask me why we need two of these, I can tell you that, but we'll get to it later. You know already, yeah. All right, so this is kind of the organization, the hierarchy of what's going on with your nervous system. You might remember back, this is kind of nice because we're kind of tying stuff in now, the stuff we did way back at the beginning, kind of refreshing everybody's memory about it. You might remember way back when we were talking about the tissues, that nervous tissue is made of two kinds of cells. The first of these was the neurons. Neurons are the excitable cells. Neurons are the ones that can have action potentials. They're the guys who get it done. Cut their sleeves off. They're the get her done guys. Right. Now, not all neurons are exactly the same. Now, this illustration is probably what we think of when most of us think of neurons. This enlarged part of the cell where we find the nucleus, this area is called the cell body. And you might remember that neurons have these little extensions of their cytoplasm called dendrites and axons. Dendrites carry action potentials in toward the cell body. Axons carry action potentials out away from the cell body. Again, we talked about that way back in the beginning. This kind of neuron is, like I said, is probably what we think of when we think of neurons. The cell body is kind of at one end of the cell. The dendrites are kind of short. And then we have this one long, sometimes very, very long axon. This type of a neuron is called a multipolar neuron. And if you're wondering how you're ever going to record that, there you go, there's a sketch. Cell bodies at one end of the cell, one very long axon, lots of little short dendrites. Multipolar neurons, for the most part, are motor neurons. They're going to carry action potentials out of the brain and out of the spinal cord and out into the muscles and glands and whatnot out there. Oops. Yeah. Yeah, the second type looks very different. For one thing, the cell body is almost in the center of the cell. So we have a really long dendrite, and then we have an equally long axon. These are called bipolar neurons. And no, they have nothing to do with bipolar disorder. There we go, there's a sketch. Okay. Bipolar neurons are found in the eyes, the ears, and the nose. In other words, the special senses.
But not the tongue.
Not as far as I know. Now the third type shown here in the picture, at first it looks like a bipolar neuron. The cell body is kind of centrally located. But you'll notice that instead of inserting itself between the dendrite and the axon, the cell body is kind of on this little stalk of tissue off to the side. This is something called a unipolar neuron. The dendrite just goes straight into the axon without having to go through or around the cell body. Sketch. Unipolar neurons, for the most part, are sensory neurons. They're going to carry action potentials in toward the brain and the spinal cord. Now, does anybody remember what the other type of cell in nervous tissue is? Oh, it's been months. And it's so late at night, Dr. Brown. How can we expect us to think about anything at 10 minutes to 9? The other type of cell we find in nervous tissue is called the neuroglial cells. Or neuroglia. Now I'm going to tell you more about it, because actually there's several different types of neuroglial cells. They do different things. I told you back then that all of them do things that will support the neurons. They're the neuron support staff. But different types of neuroglial cells do different things. Now, I don't have the picture from the Saladin book, and I don't have the one from the OpenStax book. This is the picture that I've got. There is one similar to this in the Saladin book. I don't know what's going on with the OpenStax. But this is what I've got. This kind of shows them all together. Now, in this illustration, this guy here is the neuron. right? So this is the one that's going to be having action potentials. One type of neuroglial cell is called astrocytes. These guys look kind of cool, right? Astrocytes. So what you see here is the main part of the astrocyte cell, and then it forms these little extensions with little pod feet that are attached on one side to the neuron. And then it's got another batch of these little extensions with little pod feet, and these guys are sitting on top of a blood capillary. Astrocytes are found in the central nervous system, and their job is to transport nutrients from the blood to the neuron. In particular, glucose. Neurons, like any other cell, are alive. Neurons need ATP, which means they need glucose to make ATP. Funny thing about brain cells, they can only use glucose to make ATP. Your muscle cells, your fibroblasts, your melanocytes, if they run out of glucose, they'll switch over to fatty acids, they'll switch over to amino acids, they can process other nutrients, not your brain. If your brain doesn't get enough sugar, your brain shuts down. Which is why when people have low blood sugar, like you obviously do, you start getting kind of drowsy and mentally confused and disoriented. And it's the brain that goes first when people have low blood sugar, because the brain only can use glucose to make ATP. You can't switch over to anything else. And it depends on the astrocytes to move that sugar from the blood to the neuron. It also helps to regulate calcium and potassium in the extracellular fluid around the neuron. Now, another type of neuroglial cell are the oligodendrocytes. I'll show you these guys in the picture. Now, this is the neuron, and this is the neuron's axon in gray. This guy over here is the oligodendrocyte. And you can see that it has kind of clamped itself around the axon. And there's another axon over here, and it's kind of clamped itself around that one as well. You'll notice in this picture that it's been colored yellow. Yellow is code for fat. That's right. And the reason why this guy is colored yellow is because it actually contains a large quantity of a fatty material called myelin.
It's the myelin sheath.
It's the myelin sheath. Now, oligodendrocytes are also found in the central nervous system. So these are going to form a covering, a coating around these axons that's very similar to the plastic coating we see on electrical wires. And it serves the same sort of function. It insulates the axon. Fats don't conduct electricity. Fats are very poor conductors of electricity. So action potentials can go down the axon, but they can't get out. That myelin covering is not only keeping the action potentials in the axon, but it's going to keep them from cross-circuiting from one axon to the other. Because believe me, in the brain, these guys are really tightly packed. So these oligodendrocytes are forming what's commonly called the myelin sheath around the axons of the neurons in the brain and spinal cord. Then I wanted to show you these little guys here. They almost seem to be kind of floating in space. They don't seem to be connected to anybody. And they're really small. These guys are called the microglia. And these guys, believe it or not, are phagocytic. They are also found in the central nervous system. So they're kind of there in your brain and your spinal cord. Their job is to remove bacteria, get rid of viruses, remove any cell debris, engulf it, destroy it, get rid of it. Phagocytosis. In most of our organs and tissues, where we find linings and coverings, does that sound familiar? Linings and coverings? The tissue that does that is an epithelial tissue, not in our central nervous system. In our central nervous system, you guys know about the ventricles, right? These fluid-filled chambers in the brain and the spinal cord that contain cerebrospinal fluid, they have linings. But it's not epithelial tissue, it's neuroglial cells. These guys are called the ependymal cells. They line the ventricles. They line the aqueducts. They line the central canal of the spinal cord. The job they're performing is very much like an epithelial tissue. They help to regulate the composition of cerebrospinal fluid. But even cooler, you notice these little hair-like extensions sticking out here? These guys have cilia. And the cilia project into the ventricles and into the aqueducts and into the canals. And you'll remember, again, way back at the beginning, I told you that in the human body, cilia don't move the cells. Cilia move the fluid. So not only are these guys regulating the cerebrospinal fluid, by beating their cilia, they're moving the cerebrospinal fluid. The cerebrospinal fluid is in circulation around your brain and your spinal cord. It's a very slow circulation compared to the blood because there's no heart pumping it. But all these billions and billions of little cilia keep that fluid in motion. It comes down through your lateral ventricles and into your third ventricle, and then down the Sylvian aqueduct to the fourth ventricle, and then into the central canal of the spinal cord. And it goes down the spinal cord till it gets to the end, and then it kind of comes back up the outsides and around the outside of your brain, and then it gets reabsorbed. But it's always in motion. It's always moving. Where are you guys going? All right. That's the ependymal cells. A couple more. We have some in the peripheral nervous system as well. The Schwann cells are out in the peripheral nervous system. These guys are also producers of myelin. So we're going to find Schwann cells on the neurons that are part of nerves, peripheral nerves. Again, you see them colored yellow here because they contain a lot of fat. One of the fun things about oligodendrocytes and Schwann cells is they don't just wrap around once. They actually wrap around and around and around and around and around and around and around. Layer after layer after layer after layer. Membrane, myelin, membrane, myelin, membrane, myelin. There's no electricity moving through here. No current at all. It's just an insulator. I used to tell people that the way it wrapped around was kind of like a jelly roll. And everyone would start staring at me because apparently they don't make jelly rolls at the bakeries anymore. Yeah, you don't know what they are. So someone recommended that I start comparing them to pinwheel cookies or cinnamon rolls. So, that kind of spirally around shape of many, many layers from many, many wrappings, many, many turns. Schwann cells provide the myelin sheath in the peripheral nervous system. Now, the last type of neuroglial cell, and yes, we're getting close to the end, hang in there just a little bit longer. The last one I have to tell you about are something called the satellite cells. These are also in the peripheral nervous system. But in order to tell you where they are and what they do, I have to give you a little bit more terminology. Okay. So, side trip into terminology. Our neurons are not just kind of scattered evenly around our body. They form groupings, they form clusters, they form functional units. So a group or cluster of neuron cell bodies, well, that has a couple of different names. In the central nervous system, like the brain and the spinal cord, that's going to be called a nucleus. It's gray matter that's all kind of clustered together. But in the peripheral nervous system, that's called a ganglion. A group or cluster of neuron cell bodies outside of the brain or spinal cord is called a ganglion. We also have groupings of axons. Axons that are bundled together and form a group. In the central nervous system, that's called a tract. But in the peripheral nervous system, it's called a nerve. Okay. Now I had to tell you that so I could tell you this. Wasn't there a comedian who had told jokes that way? I told you that joke so I could tell you this joke. This is not a joke, but I had to tell you all that in order to tell you this. Satellite cells are found in the ganglia. And they provide insulation there. Now, they're insulating cell bodies, not axons, so they're not myelinated, or at least not very heavily. And they regulate the chemical environment. In the ganglion. I don't have a picture of satellite cells. Somebody in my Monday, Wednesday class Googled me a picture. They look like amoebas plastered over the cell bodies of neurons. So they're just kind of like little floppy cells that kind of plaster themselves over the cell bodies. But there wasn't much, the picture wasn't all that wonderful, so I'm not going to worry too much about it, but I don't have a picture of it. All right, so we have Schwann cells and satellite cells in the peripheral nervous system, astrocytes, oligodendrocytes, microglia, and ependymal cells in the central nervous system. And you're now packing up because that's the appropriate thing to do. This is a good stopping point. We'll